All right, now that we've talked about chromosomes, let's go ahead and look at Unit 5, Lesson 2, Exploration 2. In this exploration, we're going to be looking at the process of mitosis and cytokinesis. As we talked about earlier, the cell cycle is made up of kind of three main sections. Interphase, which is that kind of just general life of the cell. Most of the cell's lifetime is spent in interphase. We have mitosis, which is the process where we divide up the chromosomes in that cell, and then cytokinesis, which is ultimately where the two new cells are going to split apart. So let's get into it. Like I said, highlighted here, interphase is important at preparing the cell to divide. In this time, we are going to copy all of the DNA, and we are going to create enough organelles for two new cells to exist at the end of the process. Here's that diagram again that kind of shows you the different aspects of the cell cycle. And again, today we're going to be focusing on the mitosis and cytokinesis part. As always, I'd highly recommend that you go through and do these activities. They're pretty easy and give you a good review of the information you need to know. Now again, mitosis is the stage at which the division of the cell's DNA takes place. All right, again, we're going to describe this process in a couple of different phases, but just mitosis in general is what we mean when we talk about the cell dividing up the DNA. Pretty cool animation there. Encourage you to check that out. I'm going to go ahead and move on. Now, there are some structures that you're going to want to be aware of that help to facilitate this process, and it's called the centrosome. The centrosome is this whole structure right here, and it's made up of a couple of different parts. First of all, there are the centrioles. Okay, the centrioles are these little cylinder-like structures. The spindle fibers are what come off of these centrioles. And so one way that I've described this in the past is kind of like a crane at a construction site. You have kind of the originator of those spindle fibers, which are those centrioles, and then the fibers that come off of it are like the cables on a crane. And what we're going to do with each of these centrioles and spindle fibers, what we're going to do with this centrosome structure is we're going to attach onto the chromosomes after they've condensed, and we're going to use these spindle fibers like cables on a crane to manipulate and move the DNA around the cell as needed, just like a crane would manipulate and move large objects around a construction site. All right, so let's look at a couple of these stages in detail. All right, at the end of this process, remember, what we're going to have is two identical daughter cells. The goal being that both of those cells are clones of each other. They have exactly the same DNA. They have exactly the same organelles because they're going to be doing exactly the same job as the original. So let's take a look. Here's a pretty good diagram that we'll use to describe this process. All right, like we said, in the cell cycle, there are three main parts: interphase, mitosis, and cytokinesis. Today, we're going to focus on these four aspects of mitosis and then the final act of cytokinesis. So first of all, we have the prophase. The prophase is the first time that we start to see those condensed chromosomes take shape. In the prophase, we go from a loose chromatin form into a condensed chromatid form. We start to see the X-shaped structures, which tells us that each of those chromatids has a copy. And we also start to see that centrosome develop with the spindle fibers and the centrioles. And eventually what's going to happen is those centrioles are going to move to opposite ends of the cell, like we see in our next picture. Once those centrioles move to opposite ends of the cell, they are going to essentially start lining up the DNA in the middle. We are going to see this characteristic line of chromatids. And the reason why they do this is to make sure that you have one solid line of all the represented chromosomes so that there's no uneven distribution of genetic material. The metaphase stage is characterized by this line drawn down the middle. Now, one thing you'll notice in metaphase is that we no longer have a nucleus. The nucleus and the nuclear envelope is gone, and all of that DNA is now free to be moved around the cell. The next part is anaphase. Anaphase is characterized by the separation of these sister chromatids. In metaphase, those sister chromatids are still connected, which is why we have the X-shaped structure there. But in anaphase, those sister chromatids begin to pull apart. And this is the first time we see the creation of two distinct groups of DNA, two distinct groups of cells. As those groups of DNA get pulled further and further to opposite ends of the cell, closer and closer to their respective centrosome, what we then start to see is this pinching off process in the middle. The cell membrane starts to reform in between these two. As you can see, the nucleus starts to reform around those chromatids. Oops. Let me go back down. And then what we're going to see ultimately is this split take place. Now, in telophase is where we start to see the nuclear envelope reform, centrioles start to go away, and then finally cytokinesis, that process splits, and we're left with two distinct daughter cells. Now, like we talked about in our last unit, it's going to be important for you to be able to tell the story of this process. I think that that's the easiest way to learn this particular material, is to think about it like a narrative. So if you get to the point where you can talk about these different pictures as stages of a book, filling in the relevant details along the way, then you're in a really good place to be able to understand how the DNA of a cell is able to be condensed, divided, and ultimately split up between two new cells. Now, let's talk about a couple other things as it relates to asexual reproduction. Asexual reproduction is any type of process that only involves one organism. So what's typically happening is the cells of a multicellular organism, or the entire organism, if it's a single-celled organism, is going to be going through this process of essentially cloning itself. When there's only one parent, you are creating an identical copy of the original. Now again, this is how bacteria reproduce, this is how single-celled organisms reproduce, and this is also how our cells are able to reproduce to repair themselves. This was one of our driving questions at the beginning of our unit, is how do cells repair? And it happens through this idea of mitotic reproduction, where if there is a gap in some tissue, if there is a limb that goes missing, if there is space that needs to be filled in, the surrounding cells are able to undergo mitosis to fill in that extra space with new cellular material. And so this is a key idea because it goes back to one of our driving questions. And it shows up in a lot of different ways, as you can see here. But the key is, this is what we're talking about when we talk about how our tissues are able to repair and replace when the cells are damaged or missing. All right, that's it for this exploration. We'll see you in the next one.